So Emma invited me to kind of showcase a bit of the work that we're doing, and the microbiome work is for patients with head and neck cancer and precancer, mostly squamous. It's an expensive project because the computational diagnostics from the information that we find from our samples generate trillions of bytes of data, so it's a lot of money to process that. We haven't at this point included patients with salivary gland tumours in the project, but it's an ambition of mine in future to get a small packet of 10 or 20,000 pounds from somewhere and include those patients too. So today I'm talking about the human oral circulating and gut microbiome in head and neck cancer and pre-cancer patients. And these are the collaborators, including some medical students who recently were involved in updating. So I'm based in the Queen Elizabeth University Hospital, Glasgow. It is the biggest hospital in Europe by bed numbers. The locals refer to it as the Death Star. You can kind of see why. I work over here in the Institute of Neurological Sciences, which is where the Glasgow coma score was invented in 1974. So, famous place. In terms of the microbiome then, there's an increasing literature identifying the role of systemic and oral commensal microbiota in cancer formation and inflammatory diseases. The human gut cancer occurs where the bacteria are abundant, which kind of makes common sense. So you get it in the esophagus and stomach, not really in the small bowel, and you get lots in the large bowel. In fact, when it comes to colorectal cancer in the large bowel, you find bacteria associated with those that are never otherwise in the human gut, and they come from the mouth. So there's some fascinating things going on and to find out about. And in particular, the pathogens that cause periodontitis or gum disease have been identified in carcinogenesis. So I've been attending microbiome meetings, and because I'm the maxillofacial guy, I've got a dental degree and a medical degree, CFRCSs, etc., etc. So I was the only person that stuck a hand up saying, so if those bacteria are important in your bowel cancer, have your patient got any teeth? Because if they've got no teeth, there's no environment in the mouth that those bacteria can be in, which I'll explain in a bit as well. And they look at me in a kind of confused fashion, what are you asking about the teeth for? I'm like, oh, do you know, anyway. So it could be that having bad teeth is really bad for you. So in terms of the oral microbiome, there's not a great deal of consistency in the literature at the moment, but some of these, and I'm putting the detail up to reassure you that people at Rob and me do work hard on science as well as medicine, but the detail's not that important. You'll all do fine in the exam at lunchtime, okay? Anyhow, so Porphyromonas gingivalis, for example, are native to the oral cavity, but you find them in other digestive tract cancers. And there's some evidence that they make the situation worse, really. Fusobacterium nucleatum is right in the crosshairs of badness, really, and so targeting that one. So some mouse experiments, we can show that an oral squamous carcinoma in a mouse model progressed worse when those bacteria were around. So dysbiosis is when your microbiome's bad, really, and that coordinates with an awful lot of different disorders. And there are also some bugs that appear to be kind of predictive from progression, so there's clearly some huge influence of all these bacteria in and around your body. There are more bugs in your body than there have ever been people alive, is one of the statistics, you know. Now going back to infection and cancer, we've known forever that HPV, hepatitis B virus and hep C virus, you end up with a hepatocellular carcinoma. Human papillomavirus is very topical. Cervical cancer might be eradicated in Australia for women because of vaccination programs. So that virus is necessary but not sufficient for cancer to develop. But if you get rid of the virus, it doesn't happen. And it's big news in the head and neck too, because so many oropharynx cancers, and not all of them though, are driven by it. In terms of other bacteria, Helicobacter for a long time has been known about in gastric cancer. And a key thing is that your microbiome is plastic and modifiable. So if you eat 30 different plants in a week, Tim Spector will tell you that it all gets much better. You can buy supplements. There's a new supplement bar that arrived in my email today, making life different. And so back to the gum disease thing then. These periodontal bugs are definitely implicated in colorectal carcinomas. There's a few references there. And it's this wee pocket where there's gum. It's not working on that screen. This will work. No, it doesn't. Okay, so the bit where the gum is kind of falling off from the side of the tooth is a low oxygen environment, and that's where these bad bugs can be. So once a patient's rendered edentulous, which is much less common than it used to be in Glasgow, there was stories about a wedding present was all your teeth out and a set of dentures, or maybe for your 18th birthday. But that's in the past now, everyone. But that's where these bugs will be. So it could be they're very significant. I've done my device. But more in the background, there's a whole international consensus, going back a wee bit now, with expert panel review. And it's not just as simple as bad bugs are there, so you get cancer. It's considered to be part of a tripartite interactome between those things that are around, other environmental factors, and a genetic and epigenetic susceptibility on the part of an individual patient. So all these things are existing in a continuum, but the question really is, would we be able to impact that and reduce the risk of cancer developing or prevent it coming back again? So traditional risk factors for head and neck cancer, and this is a part of an explainer about why I got involved in the project, are tobacco and alcohol exposure, especially if you do both. But here's an example of a lady who rocks up to my clinic, and you can see the side of her tongue doesn't look quite right. And then when I exploit the same thing that the PET scan does, slightly indirectly by putting potassium iodide on there, the Warburg effect is why cancer cells light up. So cytosol glycolysis is upregulated 10 times, and that's why the glucose gets burned up, although not an adenoid cystic particularly, is the point we were discussing earlier. So when the Warburg effect has happened, the glycogen is missing from the bad cells, and so the pale bit is all the bad stuff. And you can see there's much more pale bit than there is visible in the other slide, but the point about this lady is that she's a non-smoker, a non-drinker, and it's lateral tongue subsite. And when we did the biopsy, they said moderate dysplasia, so medium precancer change. That group is a high risk for transformation to cancer, and if you don't do anything about these women, half of them are getting cancer within 2 years. So something else is driving that. That's not a common sight for mouth cancer, and she's not smoking, not drinking. So there's clearly something driving that process, which made me want to get involved with understanding whether something else is causing it. Etiopathogenesis is a fancy word for what causes it and how it happens. And the microbiome is right in our crosshairs. So 35%, in fact, of our female presenting mouth cancers in Glasgow are non-smoker, non-drinker. It's a huge big increase. And also the sex difference used to be very much a male preponderance, but now it's almost 50-50 men and women. So our aim for this project is to identify distinct patterns of microbiota diversity between patients with pre-cancer patches and actual cancer in their mouth, and compare them to the healthy population, because we got that. We got it for renal patients and with diabetic patients in the west of Scotland, so we can compare like with like. So, could dysbiosis be a significant driver, is the question. I'm going to divert a wee bit into salivary gland cancers now, because, as we touched on a wee bit already today, a rare group of cancers, the most diverse, different set of histology types of cancer of any body site, all making it very challenging. And I reflected already that 4 to 6% of head and neck cancers mean that in Scotland every year there'll be about 50 or 60 people maximum who get one, and they're all in different places and all in different types, so it's hard. In terms of what causes salivary gland cancer, this is kind of what we find when you search for it. So there are some genetic factors and gene tests that we do if a poorly differentiated tumour lump we think might be mucoepidermoid, and that's the MAML2 rearrangement, and other fancier ones. Radiation is definitely involved, so survivors of childhood cancer quite often go on to get salivary gland tumours as well. We think maybe some things in the environment, but that's fairly thin evidence, truthfully. Some chronic inflammatory things. So inflammation doesn't cause cancer, but we consider that it's permissive for cancer to develop because the way it winds your cells' lifespan down, specifically the telomeres and the ends of chromosomes, which is what my PhD was all about. And there definitely are some influencing lifestyle factors. And smoking, if you want to check if your patient has given up smoking or not, then you do a salivary cotinine test, because the stuff comes to the saliva and it's concentrated in salivary glands. And Warfan's tumours are the second most common type of benign lump you get in a salivary gland, and they're almost exclusively happening in smokers, and sometimes a very not fit smoker person I don't want to do an operation on. If I can really cajole, encourage and bully them into stopping smoking, the Warfan's tumours go away. So things are driving processes within salivary glands that are concentrated from the things that you're eating, drinking, and maybe the bugs that are around your body too. So here's the latest iteration of all the different types of diagnosis that we'll come across. But there is a kind of big 3 or 4. So mucoepidermoids, they can be high grade or low grade. Adenoid cystics, we've talked about a lot today. And then adenocarcinomas. Quite notably in my practice, until recently, that one there, polymorphous adenocarcinoma, used to be called polymorphous low-grade adenocarcinoma, but it got an upgrade because so many misbehaved over time. And Rob and I have lots of experience of challenging situations. In fact, there's a wee lady who's not very fit under my care at the moment. Lump out of lip, they said it's a PSA, which is the benign lump. Now this new lump appears in her neck 9 months later, and they've gone back to the first lump and had a good look over it again and got fourth and fifth opinions about it, and they think it was that in the first place. Not that it would have changed what it did for her, given her age and general health. So, here's the plan for the project, okay? So patients are recruited from the clinic and asked if they'd be prepared to take part, and all they have to do is give us 3 samples. One is a swab inside the mouth of the pre-cancer patch or the cancer itself. One is an extra, we only need 1 ml actually, but we send 5 ml of blood, and bacterial ribosomal RNA is sequenced from that. And they take home a wee kit and bring back a stool sample. And then we do all the lab analysis, and that's the expensive part. Genetic sequencing really shot through the roof. And Craig Venter, the chap who pioneered a lot of it in the early 2000s, has just died actually. He's kind of egomaniac American fella, and he sequenced his own genome first, you know, and you think, oh, Craig, you know, no. I wouldn't have done that. I'm sure Rob wouldn't have either. Anyhow, and then we correlate and compare. So none of this is ever going to be direct causation or proof of such, but it can be correlation on a grand scale. So the stool sample situation is quite interesting. I get sent home with a wee one of these and they're told just to scrape off the paper. And the scientists, who are not doctors, first of all gave us takeaway tubs with those inside them. I said, some of these old buggers are going to come back with that full. Do you know what? And they actually were suggesting that you poop into the takeaway container and get a sample from it. So you can just imagine this sorts of things that are going to happen around the city. So it's meant to be just a scrape. I did have one old fella come back with that thing full to the top and the lid squidged on. Anyway, there you are, you know. Something about, I do a lot of things in my professional life, usually with gloves on, but when somebody hands you a wee smelly poly bag, you think, okay, right. You wash your hands after that, I promise you. Okay, so a quick scoot through the patient characteristics. We've screened about 180. Almost nobody turns this study down, and this is something that I really quite like in my practice. Because especially as a surgeon and doing big operations for people, which are really very daunting, and most Fridays I'm consenting the Monday patient, I hear myself explaining it to them again, and I think, oh, this sounds horrific, but we are very good at it and we do keep people safe. You kind of got one choice if you have a cancer diagnosis in your head and neck, and the choice really is you get on the roller coaster and hold on to the bar with white knuckles and just go with it, or opt not to have it. But when we ask you about a trial, you've got a choice. And people often respond very positively to that. So, will this help me, doctor? Well, it might actually directly help people, this one, but it certainly will help other people. And so there's a bit of autonomy handed back to people, I always think. And they're giving something back to medicine by taking part in these studies. So we've got 171 patients consented, and these data are from the last go through of 165 of them in terms of the mean age is that. The male and female ratio is kind of close. A third of them are cancer only in the diagnosis, about a third are just pre-cancer, and we're at the point of almost comparing those groups separately into the normals. A bunch have got both. In terms of non-drinker, 40%, non-smoker 31%, but the non-smoker non-drinkers only 10% so far and only 17 patients, so I push on to get enough of those to compare. So briefly graphically, men and women in the circle at the top, just about the same. And then dysplasia and cancer by sex and risk factor. You can see on the bottom chart, the grey line is women and the orange one is men. So the no smoker, no drinkers, more common in women, and that reflects the rest of our practice. It's always a tricky thing to assess this, you know. So I'll get you all to the thought experiment just now. If you drink alcohol, how many units do you think you have in a week? Everyone estimates it low. I myself, I'm just back from a medical conference in Paris, so I couldn't possibly let you know how that's been. And I'm sure my intelligibility got better with every passing verre de vin rouge. But anyway, so it's hard to get people to estimate that accurately, okay? But we just know if they do or they don't. Now, unlike historical dental health around the west of Scotland, now 82% of my patients have got some teeth. And uniquely, I think in the literature, we are actually doing a basic periodontal exam. So I have a dental degree and an FRCS, and I examine their periodontitis status as part of it. And nobody's got perfect gum health in this series. They've all got at least BPEs of 2, which means there's evidence of gum disease around their mouth. Not terrible, bleeding and probing, calculus and plaque. We then collect the blood, oral and fecal samples, and they're stored blood in the, so it'll go in the fridge, and there's a special freezer for the fecal samples. We tell patients if it's the day that you're bringing your sample, just put it in the poly bag. If you've produced it the day before, please put it in a few more poly bags and put it in the fridge. And I'm not putting that in my fridge. so we encourage same day sampling. So I'm going to show you now, because the data we're getting from this actually is quite sensitive and confidential, because if we get on the track of some bugs or a species of bugs that are significant, then to push forward with that research would require a patent to make a bacteriophage, and that would need big funding. So it's all got to be a bit secret and not give away to the rest of the world what we think we're onto. So I'm showing you the sequencing from the first 30 cases to give you a flavour for it. But truly, when we generate the bacterial DNA from the bloodstream and from the gut and from the mouth, we get trillions of bytes of data, so it's hugely expensive to get these computers to fish through all this. And what they're doing is getting different sequences of the RNA, and the computer then can say, right, that's spirochete, that's fusobacterium, and all the rest of it, so chunks like that. Then we get it to filter down and tell us the 25 most abundant, because they find hundreds and hundreds of different species. So, and this is a kind of nice picture chart. We're looking here on the left at the blood sample, and then the oral swab is in the middle, and on the right is the gut sample. And what you can see quite helpfully, and this is a kind of first in the world thing, is that the blood and the gut thing really tie up. So what's in your tummy does end up in your bloodstream, you know. So those are not intact bacteria. So in the hospital, if someone's rigoring with terrible temperatures that are spiking, that sort of live bacteria in the bloodstream, we do blood cultures for that. This is evidence that they have been in your bloodstream and they're there all the time, and that's what your spleen's for. It filters out encapsulated organisms, so what you put in your mouth ends up all through you, you know. It's quite interesting. And the oral cavity swab is quite different. So we now get into some statistical things, which honestly make me go a bit cross-eyed. But on the left, the AMR analysis is showing that all of our samples are dysbiotic in the cancer patients and pre-cancer patients. Betaine biosynthesis is a measure of cellular aging. So as cells age, they're far more vulnerable to transformation of the cancer. And the bacteria associated with betaine biosynthesis are again far more prevalent in our cancer patients and pre-cancer patients. And then the chemical carcinogenesis, reactive oxygen species generating bugs, again, are more common in our dysbiotic head and neck cancer population and pre-cancer population. So when we do what the non-doctors call a metadata analysis, what we're broadly showing here is that other stuff that goes wrong as you get older, like your performance stating going off, or having high blood pressure, or having had a heart attack, or type 2 diabetes, is all more common with dysbiotic microbiome. If you ever had an immunosuppressive drug for a transplant or something, that comes out too. Consuming alcohol is significant. And interestingly, in this sort of small snapshot, it's definitely the case in medicine that when you quit smoking, your risk of things goes down with every year that goes on. But unfortunately, the dysbiosis we found was still present even if you're an ex-smoker, so there were some permanent effects, it would seem. So not smoking at all is the plan, obviously. So more complex stats now. It's essentially the same message. So environmental filtering on the left, quantitative process estimates for bad bugs in the gut, blood and mouth of our cancer patients compared to normal controls. And stochasticity ratios, that's a fancy word for randomness. Essentially, it's bad to have a bad microbiome, and it correlates with dysbiotic oral cavity. So we do have some candidate species, which I'm not allowed to tell you about just now. And the great wish would be, how can we manipulate that for a benefit? So this is the conclusion so far. The mouth cancer and pre-cancer people have dysbiosis in their microbiome compared to healthy controls. And that might be driving the situation as that tripartite interactome between that, other environmental things, and your own genetic basic deck of cards, really. So the future then, kind of winding up now. In terms of future therapeutic options, it won't be a vaccine or an antibiotic. If someone has antibiotics, we don't sequence their gut microbiome for 4 weeks to make sure it's regulated itself again. It won't be food supplements either, I don't think. But the exciting thing on the right is bacteriophage therapy. So a bacteriophage is like a wee virus for bacteria, and that's how virulence factors like resistance to antibiotics is passed around the bacterial population. So in my PhD, I transplanted genes in and out of cancer cells to upregulate this enzyme telomerase to prove that made radiation treatment work much better in the cancer cells and spared the normal. And so I was able to make those with the guidance of the scientists I was working with, and we could just change cells by squirting these onto the tumour cells. So the same thing would apply. So my great wish would be that after you've had a big resection for a tumour, and we've found dysbiotic microbiome and bacteria X, Y and Z are there, that we'd be able to say, so you've had the operation, back for follow-up, you've been to see Dr. Metcalf, had the other treatment, let's cross our fingers and scan you and hope nothing happens. We would, instead of doing that, saying, and here's the medicine to reprogram your microbiome and stop it happening again, would be the plan. So that's kind of that really. A few key references here. This is our publication at the bottom, identifying our non-smoking, non-drinking, particularly female patients as a new and significant group. Getting cancer in different places in the mouth as well, not the traditional spots where, if it's a smoker-drinker cancer, where the saliva pools is where you tend to get it. So floor of mouth, retromolar trigone, and that's because the ethanol is dissolving the flat chain hydrocarbons from the smoke and accessing the mucosa there, on the one hand. Another thing is thermal trauma from smoking. So the heat and the damage from the chemicals allow access for the alcohol. Interestingly, ethanol, C2H5OH, is what's in alcohol that gets people drunk. That's not a carcinogen. It's not a carcinogen. It's the other stuff that's in amongst, okay? And they're called branched chain congeners. So the related molecules that are part of the distillation and brewing process, but that's what causes cancer. And you get most of that in home brewed or home distilled things. So there was pockets in France where they're making Calvados, or where they make poutine in Ireland, you get much more cancer because of the other bad chemicals in amongst. So I'm going to acknowledge then the other people who are collaborating. Paul Shields is a lead scientist and a great friend of mine, and the rest of the scientific team and some med students and my other colleagues who've recruited patients. I'll finish off with that slide. This is one of my favourite slides. This is from a paper in 2015 by Wusrick et al. And as ever, head and neck cancer is kind of late to the party, but for every type of disease and every body site, we know that the centres doing the most research get the best results. And this is the proof positive for head and neck cancer, okay? And there's 2 lines in those charts, okay? On the left, it's overall survival from cancer. On the right, it's progression-free survival, like I mentioned earlier on, so you've still got it, but it's not doing any harm, it's not getting any worse. And the blue line is the centres that recruit the most patients. And the yellow line is the ones that don't recruit very many. But the line that's missing, of course, is the places that don't recruit at all. And there are lots of hospitals like that around the United Kingdom and around Europe, really. So research activity and outcomes is hugely important. So the big research drive in Salivary Gland Cancer UK is quite internationally outstanding and fantastic, I think. So, and I'm very delighted to be part of it. And I promise to up my game, Emma. Right. I think that's it. Okay, thank you everyone for your attention. We're happy to answer any questions.